lovely and warm good morning to our most valued and worthy dignitaries, management committee staffs, sponsor AICTE, respected participants from all over India, and my beloved colleagues. I, Rajaram Das, Assistant Professor from Roland Institute of Pharmaceutical Sciences, on behalf of entire organizing committee, welcome you back to the third day of Series 3 online FDP program sponsored by AICTE and organized by Roland Institute of Pharmaceutical Sciences. To mark this event, a very traditional beginning, let us begin with the virtual presentation of lamp lightning and Ganesh Vandana.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
With the blessings of Lord Ganesha, let us start the onset of today's program. Dear participants, today we are having our first eminent resource person with us. Before inviting Sir to share his valuable talk, it's my privilege to introduce today's resource person, none other than Dr. Bibhu Prasad Panda sir, to all of us. Dr. Pandas is now currently serving as an associate professor in the Pharmaceutical Biotechnology Department at the Department of Pharmacognosy and Phytochemistry, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi, India. Sir is having a keen interest in nutraceutical and bioprocesses. Dr. Panda is having a vast span of 19 years of research experience in nutraceutical and bioprocess development. Dr. Panda has grabbed a total of 7 research grants from DST, DBT, UGC, ICMR, which is having an approx value of 2.5 crores. Dr. Panda has also been awarded with Rapid Grant Young Investor Award from the Department of Biotechnology, Government of India in the year 2010. Sir is also the recipient of Dr. P.D. Sethi Annual Awards for Best Chromatography Paper sponsored by Enchrome India in 2011. Sir has also received India Innovation Initiative Award for Bioreactor Design from DST, CII and Agilent Technologies in the year 2011. Dr. Panda is also having one non-exclusive international technology transfer of fermentation technology for the production of astaxanthin from the East Favia Rorzheimer to Chinese industry, that is Beijing Global Biological Corporation, Beijing, China, with a value of $50,000 in the year 2017. Sir has having a total of 76 international research publications, 2 patents on bioreactor and plant metabolites. Sir has guided till now 8 number of PhD students and 37 PG students, both from the MTech and MPharm department. With this brief introduction, it is my privilege to invite Dr. Bibhu Prasad Panda sir on board to share his valuable talk with us.
Thank you. Thank you. Hello. Yeah, thank you for giving introduction about me. Right. Actually, I'm ex-students of, you may call it alumni of Roland. Right. I actually passed in the year 1998. Okay. And then after that, I carried out my M.Pharm after clearing the. At that time, GATE was there. Right. And then I got the score of around 94.76 percentile. And then after that, I joined my M.Pharm in Bitmesra, Ranchi. Then after that, I done my M.Pharm in pharmaceutical biotechnology. Then after that, I also joined in CPS Mohuda, right, to do my some of the work there. Right, and then after that, I joined in IIT Kharagpur. Then I worked there for 2 years in research project. Right. Then after that, I joined in Jamia Hamdard in the year 2004. And my area of research is particularly on carotenoids, on nutraceutical molecules, particularly through fermentation process or bioprocess. That means the target is that how we can develop the nutraceutical molecules or phytopharmaceuticals or phytomolecules by using the bioprocess means. That means how we can use the microbial cell to produce the nutraceutical molecule, whether it may be a plant, right, whether it is directly from the microbial source. Sometimes these molecules are highly valuable, right, but we are always we are ignoring the microbial process and how and there is a need here, right, as a pharmacy profession, right, we have to work on the new molecules, new nutraceutical ingredients, and how we can generate some of the important molecules, and how we can do formulation developments and dosage form, and how we can study about these molecules, how they are actually active inside the system, in our systems, by doing in vitro or in vivo activity, right. This is the now the area, right. But in India, actually, why I'm telling you these things are that but in India we are just we are depending on the other countries, right. We are importing a lot of molecules from outside India, particularly Chinese industries, okay, European industries, and mainly from the Japan, Korea, right. We mostly import these nutramolecules, right. And actually we have capability to produce these molecules in India, but we are lacking in these things. And now there is a time, right, we should depend on ourselves, right. We should produce these molecules inside India indigenously. We should prepare, right, and we should meet our demand, right. If we depend on other countries, right, we may face problem in future, right. This is happening now also, right. We are depending on more on the other countries, right, for the nutraceutical molecules, or if you consider API also, active pharmaceutical ingredient, still we are not having the technology or technical things or know-how, right. We study a lot, right, regarding the chemical molecules processing, everything we know, right, but we are not having the exact technical know-how for making the molecules. Think about remdesivir. Now here also we are not having this. We are now yesterday only I got news that government of India has agreed to relax the import of remdesivir. Why this is happening here in India? Because we are having the knowledge, but we are not utilizing our knowledge perfectly to the 100%, right. And that's why my area of research is always towards the nutraceutical molecules, and how we can produce the ingredient, and how we can have a self sufficient, right. That means we'll not depend on the others, right. We should develop our own molecules and we can meet it, and then after that we can export the extra molecule also to outside. Okay, this is the need, right. And that's why in Jamia Hamdard, actually in our laboratory, that means in microbial pharmaceutical biotechnology laboratory in Department of Pharmacognosy Phytochemistry, Jamia Hamdard, actually we develop a lot of molecules till now. we carried out molecules on having this anti-cholesterol activity, anti-hypertensive molecules. Also, we developed like monacolin and oncoflavin, monacin, vitamin K2, menaquinone 7. Right, and also we developed vitamin D2, ergo calciferol, ergo cholecalciferol. Also we developed, right, and also we are doing research on different type of plant molecules, like how we can convert some of the soya isoflavone into egg-like forms, right, and acequols. Okay, a lot of molecules, right, are there, and there is a scope here, right, and we all therefore there is a need that as a pharmacy professional we have to do research, we have to develop new molecules, and then we can have some method to produce these nutraceutical molecules. And also there is a revenue generation also to the academic. Means, for example, if you are working in an institute, in a university, right, we can sell the technology to a pharma industries, right, and in terms we can get the money also, royalty also we may get, right. You may get also one-time payment also, like whatever we have done regarding astaxanthin. I will discuss all the things, okay, in my slide, right. I will discuss more in detail about this molecule. This is one molecule that actually I want to discuss today. And here what I want to tell you is that it is not the revenue the country will benefit, right. Indirectly also we will also get benefited. That means the worker, the innovator, right, and the place, that means the institute where you are working, right, they will also get benefited. That means the revenue sharing will be there as per the IP regulation of the institute, right, and there will be a win-win situation for all of us. That means to the public, to the society, to the country, to the industry, right, to the innovator as well as to the institute, right. Therefore, always try to focus on development of new molecules, new ingredient molecules, and the process, how we can do, how you can produce the molecules, okay, and then only will. But here in case of astaxanthin, what I want to tell you is that we started the research in but it required patience, right. It is not that in 1 year or 2 years you will get the molecules, okay, and you can process it. It required years and years of research, right, and hard working to get one process, to get one molecule. Now we started the work on astaxanthin in the year 2007. That means at that time astaxanthin was not a molecule, means not known to public, okay, as a good molecule, right. But there is a need of antioxidants. Always there is a need of antioxidant. At that time the market was filled with beta carotene, right, and lycopene, beta carotene, or vitamin E, vitamin C, like that. Number of antioxidants are there. But at that time we thought that yes, this is the molecule we should target, because at that time only 2 Israel company are producing astaxanthin, and another Japanese company, Astarreal, Fujichem, right, and Astarreal, the brand, they are actually the manufacturer of astaxanthin. Then at that time I thought that yes, this molecule having a potential, right, we should work on these molecules, right. Then we started our work on astaxanthin, and then slowly we developed a microbe like yeast, that is Paphia rhodogema. I will tell that these things in my slide. And then after that we developed the yeast, how we can improve the astaxanthin production through fermentation mode, through the bioprocess mode. And then after that actually we process the downstreaming process of the astaxanthin. Then after that we find out that yes, this molecule having a good in vitro activity and in vivo activity. Then we developed the formulation of these astaxanthin molecules. Then after that I asked a lot of Indian industries to take the product to process through the technology transfer means. But no Indian industry at that time were aware of astaxanthin, that yes, this molecule is actually having potential, future potential, and everybody denied. At that time I can name the company AICTE, Reliance, Sun Pharma, right, Cadila, most of the company I approached, right, but they denied at that time. And then finally I approached the multinational companies like Chinese company, right, Beijing Global Biological Corporation, Then they approached me, and then they taken my technology, and they are now producing astaxanthin 7 ton per month, right? That means you can imagine that what is the volume they are producing now, and they are getting commercial benefit, right? And they are also selling to European countries, right? To the feed manufacturer, to the nutraceutical industries they sell, and to India also they sell. That means this is the things we should know, that what is the value of research, right, in nutraceutical ingredient. Not only nutraceutical ingredients, we in API, active pharmaceutical ingredients, so basic API you are unable to produce. Antibiotics, you can imagine, vitamins, right? Vitamin B12 also we are unable to produce in India, right? We are unable to produce vitamin K2, menaquinone 7, right? Only one industry is able to produce vitamin K2 MK7 in that is Viridis Bioscience, right? Vitamin E, vitamin D, we are still there is a problem. Only one Fermentabiotic is producing, no any other company, right? We are fully dependent on the Chinese company, right, and European company. Therefore there is a scope here, right? Why there is a need to do research, more and more research, and there is a good demand, right? And always you will be success in your research, right, if you do hard working and in patient, right, in patient's manner, right? Then you will get definitely result will come, and you will get also financial benefit also. Name will be there and financial benefit will be there to both to the innovator as well as to the institute. Therefore, and today my topic is related to astaxanthin, but because I cannot tell all the molecules whatever we have developed here, but in the later on I can discuss all the molecules that we have developed or in our hand, right? And they are also actively under consideration by the pharma industries for the technology transfer. But today I am here to discuss about the astaxanthin. This is a real antioxidant molecules. Whatever we have seen, vitamin E or beta carotene or lycopene or any of the things, natural carotenoid, whatever we are seeing, right, or antioxidant, astaxanthin is the king of the carotenoids. That means is it the natural super carotenoids, right? And before that, I want to discuss what are the carotenoids, because all of you know what are the carotenoids, what is the need of doing research on the carotenoids. And here, if we see the carotenoids, there are two different variety of the carotenoids we find in the nature, right? One is oxygenated carotenoids, another is non-oxygenated carotenoids, right? Non-oxygenated carotenoids, that is carotene, okay, particularly we know lycopene and beta carotene. Okay, these two are the major non-oxygenated carotenoid widely used in the pharma industries, in the food industries, in the nutraceutical industry, in the cosmetic industries, right, in the feed industry also. And if we see the oxygenated carotenoids, they are called the xanthophiles, right? And major xanthophiles are zeaxanthin, lutein, right, beta-cryptoxanthin and astaxanthin. Now, out of these xanthophiles, you can find that zeaxanthin and lutein are widely used in the eye health. That means in the ophthalmic preparations, right? There is a good demand of lutein and zeaxanthin, and now also we are focusing on the mesozeaxanthin, okay? Because if you see the eye health ophthalmic preparations, till now we don't have the eye drops, right, ophthalmic preparations containing mesozeaxanthin, zeaxanthin and lutein, right? And there is a scope here, right? That means we should work on the formulations of the carotenoids, right, like lutein and zeaxanthin, how we can prepare these xanthophiles so that we can directly deliver into the eye, right, through the ophthalmic drops, right? Still there is a scope here. That means I want to tell you here that oxygenated carotenoids, that is xanthophiles, are having very high value things, right, like lutein, zeaxanthin and mesozeaxanthin, that is not mentioned here. But these three are very important xanthophiles, particularly known to public, also known to consumer also, that they are very good for eye health, right? And in the xanthophil also we have astaxanthin. Astaxanthin is not good ophthalmically, it is not actually helpful, but it is an antioxidant, very good antioxidant, which can protect the cardiovascular health, right, and neuroprotective effect, right? It can prevent the cancer, it can protect the lungs from the damage, right? And also it is a very good antioxidant for the athletes. That means because it carry the oxygen, it protect the cells, okay, from the external damage. And also it is a good cosmetic ingredient also, because if we suffer from UV radiations, right? That means if there is a skin burn is there, right? If we are taking xanthophyls like astaxanthin, it can protect the skin, right? And then after that, we have also other carotenoids like new carotenoids. You can work on those carotenoids like phytoin and phytofluin. They are upcoming carotenoids and most widely used in the cosmetic industries, right? Because they are white carotenoids, white and yellowish white carotenoids. They're not colored, right? And they find wide application in the cosmetic industries and we have a scope there, right? The phytoin and phytofluin, and but we'll more focus on the astaxanthin today. Now, these are the structures. I already discussed that different type of carotene and xanthophyll, right? You can see hydroxy group, right, in the xanthophylls, right, in the lutein, zeaxanthin, beta-cryptoxanthin, astaxanthin, and fucoxanthin. I want to tell you here that fucoxanthin is a very good carotenoid, upcoming carotenoid for anti-obesity. That means we can control the obesity or we can control the weight. We can good weight management we can do by using this fucoxanthin carotenoid. That means you can imagine that there is a scope here, right? The fucoxanthin, right? And you can see here this phytofluin and phytoin. Okay, these are the white carotenoids, right? And find wide application in the cosmetic industries. Okay, but in India, we will not find any company producing these two molecules, right? Only Israel company, they are Lycored and IBR. Actually, they are producing phytoin and phytofluin, right? And this beta-carotene is most widely produced in India, but still we are dependent on Ukraine and China, right? We are importing a lot of beta-carotene from ZMC, right? That is a Chinese industry. They produce beta-carotene in large scale by using fungi, that is Blachycelia trispora, right? And they produce beta-carotene, and that we import and we use in our food industries, in pharma industries. There is a huge demand. But alpha-carotene and gamma-carotenes are still under development, right? And we don't have that much scope here, alpha carotene and gamma carotene. But lycopene is again another molecules which find wide application as antioxidant, okay, for eye health, right? And particularly we get lycopene from the tomato, but still we can get the lycopene from the fungal source, that is Blachycelia trispora. And beta carotene and lycopene are obtained from the same fungus, Blachycelia trispora, right? And the China is the world leader in producing these two molecules, and we are importing these molecules. And there is a scope here to produce beta carotene and lycopene from the fungi like Blachycelia trispora. We tried, but we failed, right? In last 2 years, we tried to produce beta carotene and lycopene in our laboratory through a grant, a DST grant, but we are unable to produce because the fungus Blachycelia trispora is highly unstable and is not able to give good amount of beta carotene. Therefore, we stopped that research, right? But still, there is a scope to enhance or to produce the beta carotene and lycopene by using this Blachycelia trispora, right? And these are all about the structural elucidation of this. Sorry. Hello, hello, hello. एक्चुअली मेरा क्लास चल रहा है, मैं 1 बजे कॉल करता हूँ, राइट? ओके ओके ओके. Sorry, there was a call, emergency call was there, but I caught it. Right now, next important thing is that I want to discuss that what is the application. Already I discussed, right? Still I want to present here that if you see the animal feed market, right, mostly you will find lutein, canthaxanthin, astaxanthin, beta carotene, and lycopene, right? These are the most commonly used carotenoid in the animal feed, right? In the food, industries: lutein, canthaxanthin, astaxanthin, beta-carotene, and lycopene. These are the carotenoids actually wide application in the food industries, right? In the supplement industries, that means in the nutraceutical industries, in the supplement industry, the food supplement industries, right? Mainly lutein, lycopene, beta-carotene, astaxanthin, canthaxanthin, and lycopene again, right? Is find wide application in the supplement industries. In the pharmaceutical industries, actually lutein and lycopene are most commonly used, because lutein, you know, all of you, it is widely used in the eye health, and lycopene is antioxidant, okay, and cardiovascular health, right? Mostly we use lycopene. And in cosmetic industries, right, most of the carotenoids which can protect the UV light, they are used, right? Particularly astaxanthin, canthaxanthin, lycopene, and beta-carotene. Here we can add these two new carotenoids, that is phytoin and phytofluin. Right now thing is that now here there are two different forms, right, actually preferred by the industries. One is the synthetic carotenoid, another is the natural carotenoids. Now food and beverage industries particularly prefer both, that means synthetic as well as the natural carotenoid. But if you see the economy of the application of the carotenoids, now synthetic carotenoids are low cost in compared to the natural carotenoid, obviously, but natural carotenoids are always full spectrum, good activity in compared to the synthetic carotenoids. Therefore, food industries actually they use both the carotenoids as per the cost effectiveness, right? Sometimes they use synthetic carotenoids, sometimes they use natural carotenoids. In case of cosmetic industries, they do not prefer this natural carotenoid, right? Because the synthetic carotenoids are commonly used in the cosmetic industries. But in the dietary supplement, in the nutraceutical industries, they do not prefer synthetic carotenoids, but they prefer the natural carotenoid, obviously, because if we are taking it internally, right, then obviously natural carotenoid, full spectrum carotenoids are always preferred over the synthetic carotenoids because of geometrical configuration, right, geometrical isomers, right? Actually, the natural carotenoids are actually preferred by the dietary supplement industries. Now, if you see the animal feed industry also, again, same thing, right? Synthetic carotenoids are not preferred by the animal feed industries, right? They always prefer the natural carotenoids, synthetic carotenoids, because indirectly what happens here is that if we are giving a synthetic carotenoid to animal, right, indirectly we take also the same carotenoid. For example, if we give synthetic astaxanthin to in the aquaculture feed, right, in the food chain, again we will get the same carotenoid in our system also. Therefore, some of the feed, right, we should use the natural carotenoid, not the synthetic carotenoids. Okay, that means there is a scope here. That means natural carotenoids are find wide application in the feed industries, and also in the pharmaceutical industry. Also, there is a confusion here, right? Always they want 100% pure active molecules, right? Therefore, the most of the pharma industries, they use the synthetic carotenoids. They do not use the natural carotenoid. Most of the pharmaceutical industries. This is the question mark here, right? We should, that means as a pharmaceutical profession, right, we should try to use the natural carotenoid, not the synthetic one, because synthetic carotenoids having some effect, right? That means some adverse effects are there when we are using the synthetic carotenoids, because most of the synthetic carotenoids are derived through petrochemical routes, right? Therefore, we should focus on the natural carotenoids and their application in the pharmaceutical industries. Now, next question is that if we want to do carotenoid research, right, what are the things we have to focus? What are the main challenge we face, okay, in the carotenoid research, right? Number one, that is the production and purification, because this is the main first challenge we get, okay, during the carotenoid research, right? Because whatever the carotenoid you want to produce, that means whether it is a natural carotenoid or it is a synthetic carotenoids, first important is the production. That means how we can produce the carotenoid in a microbial cell, or by chemical route, how we can produce it, because it is a very complex molecule. It is not that a small molecule, right? It is a medium-sized molecule, right? And therefore we have to think, we have to do a lot of means there are a lot of challenges when we want to produce this carotenoid. The next question is the purification again, right? Purification, because purification of the carotenoid is these carotenoids are mostly, they are the non-polar molecules, right? And there is a challenge here whenever we want to purify the non-polar molecules by using green chemistry. That means without using organic solvent, how we can purify the molecules, right? How we can purify the carotenoid. Okay, that means this is the first challenge, is that how we can produce the carotenoid, how we can purify the carotenoids, right? Then the next challenge is the analysis. Okay, this is also an area of research also here. Now, if you are from pharmaceutical analysis background, right, you may be thinking that analysis of molecules, like small molecules, is very easy. You can take a C18 column, you can do HPLC, you can do TLC, right? Easily you can analyze the molecules. But if you are considering, or if you want to analyze the carotenoids, right, we have to use C30 columns, right? C30 carotenoid columns are most commonly used carotenoids columns for analysis, and particularly the mobile phase is also very difficult. Okay, establishing the mobile phase for carotenoid analysis also very difficult because of degradation. That means during the analysis, exactly, the molecules may degrade also, right? May degrade. Therefore there is a chance, there is a analysis, a difficulty in the analysis also there, right? And also whenever we are doing TLC also, thin layer chromatography, at that time also there is a chance of degradation. If the molecule is highly unstable like carotenoid, immediately it degrade, right? And you may not get the exact result. And further, there is a isomer. There are different isomers are there, okay, in the carotenoid. In one carotenoid you may find trans isomer, cis isomer, different type of isomers you will find, right? And analyzing the isomer, okay, in one carotenoid also there is a challenge here. That means the analysis of the molecule is also there is a good scope here, how we can analyze the carotenoid molecule, right? And next challenge is the formulation development, right? That means whenever you will be given a carotenoid, then first challenge is how we can make it a solubilized form, right? Otherwise it will not be, bioavailability will not be there. Therefore, first of all, we how to make it a solubilized form, how, so that it will be more bioavailable, right? And without and how we can make the formulation of these carotenoids, that is also one challenge, right? Therefore, a lot of research nowadays are there, how we can make the carotenoids more bioavailable, right? More water soluble, okay, so that easily we can take these carotenoids and make it into easy dosage form, into tablet or capsule, right? Or to a eye drop, right? Or injectables, right? How we can make it, okay? Still there is a challenge of the formulation development of the carotenoids. Then stability is another problem, right? Because carotenoid are highly unstable molecules. That means immediately they degraded by the light, they degraded by the oxygen, they are highly sensitive to the temperature. Therefore, stability issue is also a problem, right? That means there is a problem here, how we can develop the formulation and how we can enhance the stability of these carotenoids. These are the main challenges actually we do. And if we are interested in the carotenoid research, then we have to focus on these challenges. Then only we can develop new molecules, new product, and new formulations, and also the activity also. Now coming to the production and purification. Now, is the slide is clear to you? Is it visible? I'm asking you, right? If it is not clear or not visible, immediately you can ask me also.
It's visible, sir.
Yeah, thank you. Now, first challenge is the production and the purification, right? And whenever we are thinking of production, right, and purification, first of all, we have to think about the source, right? which source we have to use, right, for the production of the carotenoids. It may be astaxanthin, it may be a beta carotene, it may be lycopene, right, or it may be zeaxanthin, it may be mesozeaxanthin, it may be lutein, or it may be other carotenoids like new carotenoids like cryptoxanthin, right, other carotenoids also there. But first of all, we have to see the source. What are the natural source actually we can target, right? We can get the natural astaxanthin. We are not discussing about the synthetic astaxanthin, but because my area is not related to synthetic, right? Therefore synthetic, because I'm not specialized in the medicinal chemistry, because I always focus on natural products and how we can get these molecules, carotenoid molecules or any other molecules through the natural source. Those things I target, right? Therefore, first of all, we have to see the target, that means the source, right? Whether we should go for the plant source, whether we should go for the algae source, or we can go for the microbial. It may be a bacteria, it may be a yeast, or it may be a fungi, right, or any of the microbial source. What are the source we can target, right? That is the first important thing. First of all, then if you are targeting a source, natural source, then whether they are approved or not, because sometimes we may target a source which are not approved by the regulatory agencies. Okay, if we are targeting a source, new source, then first of all we have to see whether that algae, that plant, or that microbial is approved by the regulatory agencies or not. Then only we can select the source. If you are selecting a new source, then there is a new challenge. That means you have to first approve the microbial, whether they are grass category or they are non-grass. If they are non-grass, then there is no value of doing the research on the production, because you will not get permission to produce the product or to market the product or to manufacture it, right? Therefore, first of all, we have to see the source, right? And we have to find out the best source, right, which is able to produce in good amount of carotenoids or any molecules, natural molecules. Then if we are thinking of synthetic one, right, which form, right, whether it is producing, means if we are doing synthetic route, right, to produce the carotenoid, right, whether it is producing cis or trans, okay, because cis isomers are not preferred by the pharma industries or by the nutraceutical industries, because all of you know the trans isomers of the carotenoids are always very good. If you see the bioactivity profile of the trans isomers, they are always better than the cis isomer, right? Therefore, trans isomer, whether if you are targeting the synthetic route and you are able to produce cis isomer, then no value at all, right? Nobody will buy your product, nobody will take your technology, right? If we are able to produce the trans isomer of the molecule, then there is a value, right? And then after that, we have to think about the whether the molecule is free form or esterified form. Sometimes what happens here is that whenever we would develop the molecule, they are free form, right? Because you have not esterified it, okay? But if you are esterifying the free form of the astaxanthin or any carotenoid molecule to a fatty acid, that means if we esterify with a fatty acid, then the stability is enhanced. It is seen that whenever it is esterified, right, the stability is enhanced, but there is a demerit also. The esterified form of the carotenoid is also having low bioavailability than the free form of the carotenoids. Therefore, we have to think whether you want to synthesize a free form of the carotenoid or esterified form of the carotenoids. Now, next important thing is that which solvent we will use, right, to purify, okay, to purify the carotenoids. Whether you will go for the organic solvent or whether you will go for the SFE, that is supercritical fluid extraction technique, because all of you know that SFE nowadays is booming, right, and always there is a demand for SFE product. But if we are using organic solvent like class 3 solvent, class 3 solvents like ethanol, right, or modified ethanol, or any of the eutectic solvents, then it is okay. But if we are using a class 2 or class 1 solvent, organic solvent for extraction, then there is a question mark, right? How much solvent residue are remaining with the product. right? And then there is a challenge here also, because organic solvent you cannot remove 100% from your product. Therefore now there is a shift to that, how we can purify the molecules, how we can extract the molecule by supercritical fluid extraction technique, that is SFE, or using a co-solvent like ethanol, and how we can use the SFE method to extract the carotenoids. Okay, this is the challenge here, right? Now how we can use the better method or green chemistry for extracting the molecules. And how to extract it, because all of you know that the fourth one is very important, right? Because all of you know that carotenoid is an intracellular molecule, right? It is not extracellular, it is not secreted to outside, right? Therefore we have to rupture the cells. What method you will use, right, to rupture the cells? Whether you will use enzyme, whether you will use physical method, whether you will use any of the chemical method like harsh chemical like DMSO, or any of the chemical you are using, or whether you are using surfactants. Okay, that you have to think, right, how we can extract the carotenoid from the cell by minimizing the chemical impurities. That means we should prefer the physical method of extraction, but that is energy consumption method, right? Like you can go for dynomial technique, you can go for homogenization, high pressure homogenization technique, you can go for ultrasonication techniques, right? You can go for probe sonication or ultrasonication technique for rupturing the cells and extracting the molecule out of the cells, right? And also we can use enzyme like glucanase enzyme, we can use different type of enzyme, protease and cellulase enzyme, right? And we can rupture the cells, you can get them, you can molecule, right? But we can also use DMS or dimethyl sulfoxide, right, to rupture the cells, and we can get the molecules out of the cell. But if we are using physical method, that is always prefer. And then we have to go for the extraction by using the SFE, and then after that we have to concentrate it and purify it, then after that stabilize it. Now this is the pathway, actually the carotenoid pathway, particularly in most of the microbe, actually it is followed, right? That means you can see the pathway here, that phytoin, lycopene is there, phytofluin is there, lycopene is there, beta carotene is there, then zeaxanthin will be there, canthaxanthin will be there, and astaxanthin will be there, right? This is the pathway. Now scientists are also working on metabolic engineering. What they do actually, now if you want one molecule like lycopene in your cell, right, we stop the pathway here. That means we can use some of the inhibitors, right, and we can accumulate the lycopene in the cell. That means we can accumulate anywhere, right? That means if you want phytoin, we can stop the pathway here, right, and we can enhance the phytoin content inside the cell, right, by inhibiting the pathway. This is the area of research now, right? That means we can do some of the metabolic engineering in the cell, so that the initial metabolites that will be accumulated more in the cell, and you can get more amount of phytoin. But if we will make it free, what will happen here is that the cells will grow, right? The plants or the algae or the microbial cell will grow, and they may produce all the carotenoid, okay, in the cell, and you may get astaxanthin in higher quantity, but all the carotenoids will be there inside the cell, and you may not get one particular carotenoids in high amount. Therefore the metabolic engineering is now the area of research, how we can enhance one carotenoid in a cell, okay, by inhibiting some of the pathway, right? We can accumulate. That means if you want to accumulate beta carotene, we have to stop here. That means the cells will stop producing zeaxanthin or astaxanthin. It will produce only beta carotene, and we may get the beta carotene in higher concentration. That's why the metabolic engineering, knowing the pathway, okay, what is the flow of the carotenoid synthesis inside the cell is very important to know, right? Now thing is that the analysis part, right, we analyze the carotenoids by a different way, right? We can use visible spectroscopy method, right? We can use, but see here, visible spectroscopy is used when it is a colored carotenoid. But in case of phytoin and phytofluin, they are white carotenoids, right? We have to use a UV spectroscopy method, right? Visible spectroscopy will not give the data. That means you cannot analyze the phytoin and phytofluin by using the UV spectroscopy. We have to UV spectroscopy is used for the phytoin and phytofluin. But if you want to analyze other carotenoids, that is orange or yellow or red carotenoids, right? Mostly we follow the visible spectroscopy. This is a good method, right, to understand the preliminary things, right? If you want to see qualitative, for example, if you have a cell and you know that it is producing some of the color compounds, xanthophyll maybe or carotene maybe, but you don't know, but you don't have HPLC or HPTLC in your lab, then immediately you can start experiment, right? You can rupture the cells by DMSO, right? Easily you can rupture the cells by DMSO. You can, if you treat the cells with DMSO at 60 degrees centigrade, okay, preheated DMSO, it will rupture the cells. Then you extract it with ethyl acetate, okay, and you will get the molecules. Then after that you can just analyze by visible spectroscopy in a wavelength of around 400 to 500 nanometer. Then you will find that there is a spectrum, trimaxima spectrum you will get. If it is giving a trimaxima peak, that means definitely it is giving a carotenoid, but we have to think what type of carotenoid it is. Okay, that means preliminary experiments. If you find a molecule, a microbe which is giving a color molecules, right, then immediately you do your experiment. You can start with minimum utilization of your laboratory or chemicals, right? That is what I want to tell you. But if you have a sophisticated analysis method like TLC or HPTL, TLC you can do, manual TLC also you can do, and you can extract the molecules. You can bend, you can purify the molecules by doing the preparative TLC, and then after that you can extract it from the silica, right? And then after that you can analyze again by the UV spectroscopy, visible spectroscopy, and you can understand that what type of carotenoid it is. Or if you have a machine like HPTLC, or if you have a HPLC or LC system, or LCMS, HPTLCMS, then it is advantage, right? You can go deeper and deeper, right? You can do a lot of analysis for these carotenoids. Right now thing is that I think this slide is somewhat not clear, right? Therefore, try to visualize the slide here. Now, if you see the carotenoid, right? It may be present in the free xanthophyll form. Free xanthophyll means free carotenoid. Okay, xanthophyll means it is hydroxylated, right? This free xanthophyll, or it may be in monoester form. Okay, that means the fatty acid is attached. Okay, it may be a monoester form, or it may be a diester form. Now, these things you can analyze only by using HPLC or by using LCMS, right? You cannot analyze these carotenoid by using the spectroscopy. Therefore, if you want a deeper analysis of the carotenoid, whether they are present in free form or esterified form, right, then we have to go for HPLC with LC with MS, okay, or you can go for HPLC with C30 columns, right, and that will give you more detailed data. But still, we have a solution here. If they are not visible, if they are present in the diester form, then you are monoester form, just you can break the ester bonds, right, and you can make it to free astaxanthin or free carotenoids, and then you can analyze by the spectroscopy. That means visible spectroscopy. There is a scope also here. That means it is not that if it is esterified form, you will not able to analyze. Just to break the molecules at the that means you can free the molecules from the deesterification you can do, right, and you can analyze in the form of free, right? That is possible. That's why UV visible spectroscopy actually is preferred for the free astaxanthin free carotenoid analysis. Now, thing is that how we can analyze the carotenoid by this- by spectroscopy. That means by visible spectroscopy. You can see that trimaxima peaks. Already I told you this is a lutein molecule, right? You can see the yellow band here. This is a trimaxima. That means it is having a three maxima, right? And these three maxima are the characteristics of the lutein. That means if you have a beta carotene, then the trimaxima will be different. Okay, the height between the maximum lambda max, right, and the minimum lambda max, we can measure, right, and we can determine what type of carotenoid it is, right, and we can also similarly we can have a zeaxanthin, okay, having a different peak character, right, and also we have a lycopene, then we have a and also very important is that if there is a chlorophyll is present in the plant, right, we can also understand that where it is, whether they are present or not, because chlorophyll sometimes may hinder the lutein peak. But if we are getting a trimaxima peak, right, that means chlorophyll is there, but the lutein is also there. But these things you can do clearly if you do the TLC. Then after that you are extracting the molecules from the TLC, from the silica gel, and then after that you are analyzing the molecules by visible spectroscopy. Then that will give you a clear idea about the carotenoids. And also color is also very important. Sometimes it may be a red color, orange color, or it may be yellow color. Okay, based on that also we can identify the what type of carotenoid it is. Now coming to the geometrical isomer of the carotenoid, I'm discussing here beta carotene. I'm not discussing about the astaxanthin. I will discuss later on, right? Because this carotenoid is widely studied by us, right? Therefore, I want to discuss the beta carotene. Now you can see what is the difference between a trans beta carotene and a cis beta carotene, and why trans beta carotene are always preferred, right, by the pharma industries, by the natural food industries or nutraceutical industries or functional industries, because you see the cis isomer of the beta carotene is always curved. That means there is a band here. That means at the 15th position, right, it is band, right? If it is band at the 13, then that is 13 cis beta carotene. Okay, if it is 9, then it is 9 cis beta carotene. These are all cis isomer of the beta carotene, and they are not prepared at all. Now, if we see the bioactivity, the trans beta carotene or trans isomeric form of the carotenoid, any carotenoid, if we think that trans isomer is always better, having higher bioactivity than the cis isomer, right? And that's why whenever you are doing synthetic carotenoid, right, or if you are developing a carotenoid through natural route, now we have to understand that whether the carotenoid which is produced are all trans, or there is a mix of cis and trans, right, or it is a cis isomer. If it is a completely cis isomer, no value. That means the bioactivity of the cis isomer of the carotenoids are always less, okay, in compared to the trans isomer. Because that why it is happening here, I want to tell you that trans isomer actually relocate themselves easily in the cell. Okay, they easily penetrate inside the cell membrane, right, through the cell membrane, and they give a higher activity. But if it is a cis isomer, actually there is a difficulty in the entering of the molecules inside the cell, and the antioxidant effect will not be there. No doubt, in the chromatogram you may get a peak. In the spectral data also you may get a peak. In the formulation also it will pass. But if you see the bioactivity or bioefficacy, you will find a less bioefficacy of the cis isomeric form of the carotenoids in compared to the trans isomer of the molecules. Therefore, always try to have a trans isomeric form of the carotenoids, not the cis isomeric form. But there is a contradiction. I will show you in the slide, right? Some scientists, they are also working on the cis isomer, okay, and they have seen that cis isomer is easy to formulate, Okay, but the trans isomer is not easy to formulate into the nano emulsion form, right? I will show you in the slide, next slide, right? Wherever we are doing the formulation of the carotenoid, it is found the cis isomer is having good formulation capability than the trans isomer of the carotenoids. And this is the typical chromatogram of carotenoids, right? You can see there are a lot of carotenoids you can find, okay, in a chromatogram, right? And this is the very important character I want to tell you, right? Now sometimes what happens, this is the problem in the industry and into the academia also, right? What happens here is that some people, right, some organization, right, they sell the carotenoids as a natural carotenoid, but that contain the synthetic one, right? Because there is very difficult to know which one is synthetic, which one is natural, because the color, the property, the spectral property, everything will be very same, right? If you considering the natural and the synthetic, you will not able to understand which one is natural, which one is synthetic, which one is natural by looking at it, right? Now if we do the chromatography, that means if we do the HPLC using a C30 column, right, not C18, we are using a C30 columns, carotenoid columns, right? And if we want to elute the molecule, now you will find that if it is a natural molecules, it is not that 100% one molecule will be there. Now, for example, if we are getting a beta carotene from the carrot, or if we are getting a lycopene from the tomato, or if we are getting astaxanthin from Haematococcus algae, right? It is not that only one carotenoid will be there, okay, in the natural products. It will be a mix of other carotenoid also. That means all the carotenoid, I have shown you the slide in the biosynthetic pathway, that whenever we are getting astaxanthin, there are this pathway actually carry from the phytoin, okay, phytofluin, okay, beta carotene, all the carotenoid will be there, but the end product will be also there. End product will be always high, but other product will be also there, other metabolite will be also there. Now, if you analyze the carotenoid, okay, if you get the chromatogram, you will find that other molecules also there in it, okay, in that compound. Then that means that this is a natural molecule, right? If it is synthetic, then the chromatogram or the fingerprint will give you a sharp single peak, okay, and you will not get other molecules like lycopene, beta carotene. These molecules will not be there, zero, right? And that indicate that then first indication will be there, yes, there is a adulteration, right? That means there is some mixing of synthetic with the natural, or 100% natural, 100% synthetic. Okay, this is the means if you want to do quality control of the carotenoid, easy way you can do it. And another thing also you can see here, that if it is a synthetic molecules, you will get most of the molecules will be in the cis form, right? If you want to separate the molecules trans and cis, you will find most of the molecules will be fall under the cis form, right? Trans isomer of the carotenoid will be very less, okay, and that indicate that the product is having mixed with the synthetic one, right? And then you will understand that yes, my molecule is actually not natural, but it is adulterated with the synthetic one, because all of you know that the synthetic are very less cost, right, in compared to natural one, and always industry try to make profit, okay, out of the product. Therefore they always try to do adulteration, okay, and we have to check it through the quality control means, right? Therefore we have to understand that what is the problem and how we can solve this challenge, right? And next important thing is that if it is a synthetic one, right, or natural one, then you can go for the carbon dating. That means whether it is having isomers of the isotopes of the carbon or not, right? If it is a natural, then you can go for the carbon dating, right? You can understand that is the last step, but that is not easily done by a normal laboratory, normal pharma laboratory. You cannot do the carbon dating, but we can do HPLC, we can do TLC, we can do mass spectroscopy to understand whether the molecules, carotenoid molecules, are natural or they are synthetic or they are mixed, right? This is how, and if you see the chromatogram, easily you can understand that how actually the chromatograms are well differentiated, well resoluted if you do the HPLC. And you have to develop the mobile phase also. It is not that when one mobile phase you will get the molecule, right? And if you have a PDA detector, if you don't have mass detector, no problem, right? You can have a PDA detector, right? And you can see the spectral data of each peak, right? And you can correlate with the spectral data of the carotenoid, and then you can find out that yes, this molecule is lutein or retinol or zeaxanthin. Okay, if you don't have the marker compounds, right? Because some laboratory don't have marker compounds because they are very highly costlier molecules, right? Therefore marker compounds is very high cost. Therefore we if we know the spectral data just by doing the PDA detector, right? By doing a PDA detector, by using a PDA detector in the HPLC, we can understand that what molecule it is, right? And without using the and if you have a mass detector, then well, right, easily you can understand. And also another thing also we can see whether the carotenoid is free form or whether they are in the esterified form. Most of the natural carotenoids are present in the esterified form, right? They are not present in the free form, right? Esterified form easily gives a data, right? Because when you elute the molecules, elute the carotenoid molecule, always the esterified form delayed, the elution is delayed, right? But the free carotenoid is always eluted before. I mean, that means the RT, if you see the retention time of the sorry, is it visible? The slide is visible to you? In between I'm asking you whether it is visible. Is it clear to you whatever I'm telling you?
Yes, sir.
Or you are able to understand? If there is any
Yes, sir. Continue, sir. Continue, sir.
okay. Now, thing is that if the natural astaxanthin or natural carotenoid is there, now it is always having a esterified form. Therefore, the elution time or the RT retention time is always will be more in compared to the free carotenoids. Therefore, by observing the peak retention, right, we can also understand that whether it is a mix with synthetic or whether it is a natural, right? And these are the simple techniques you can follow, right? And we can find out whether they are natural or whether they are synthetic or they are mixed, right? These are the some of the carotenoid peak analyzed by the mass spectroscopy, already discussed. Now these are the TLC of carotenoid. You can see if it is a you can see here the free astaxanthin is at the initial, the RF value you will find here, right? Low RF. Okay, but if it is esterified, right, you will find a band here. That means it is a compound which is derived from the natural source. Now if you are getting a peak of astaxanthin diester, right, that means it is a natural carotenoid. That, but if we are getting a free astaxanthin sharp peak here, that means it is a synthetic one. That means by observing the TLC or chromatogram, right, TLC fingerprint, you can also understand whether it is a natural one or whether it is a synthetic one. Now next is the formulation aspects, right? Here actually we do a lot of formulation because formulation is very important task, right? Problem associated with the carotenoids, because carotenoids are actually lipophilic molecules, right? And if we take carotenoids, these lipophilic molecules, right, mostly they are degraded in the GIPH, right? And they are not able to absorb, bioavailability issue will be there. Okay, formulation you cannot do a proper mixing, right? And there is a issue of bioavailability, pharmacokinetic profiling, everything will be a problem. But if we do micronization or emulsification or microencapsulation, right, or nano emulsion form, right, then it is found that it is having a good bioavailability, right. That means we can increase the bioavailability of the carotenoids by mostly we do microencapsulation technique, right, and emulsion technique, okay, nano emulsion techniques, right, for enhancing the efficacy of the carotenoids. Right, now see here, this is the things actually we do, right. That means we use CO2 based extraction and nano size formation of these carotenoids like beta carotene or zeaxanthin and this astaxanthin, and by using the CO2, this is not a simple CO2 like SFE. These CO2 are very different, the machine is different, right, which is actually used to make the crystals of the carotenoid, right, into a nanocrystals. That means by passing the CO2 we can reduce the size, right, and we can prepare the nanocrystals of the carotenoid. But the problem is that whenever we do, I already told you in my previous slide, the trans isomer actually converted into cis isomer, okay, when there is a nanoparticle formation. This is the problem, but till now we don't have any solution for this conversion. That means if it is a trans isomer, right, and it is forming the cis isomer, then there is a efficacy issue is there, but it is forming the nanoparticle. Now see here, this is the problem. Now we are preparing a nanoparticle for increased bioavailability, but we are losing the bioactivity. Therefore, this is the area of research now, right? How we can keep the molecule intact, but we can get the nanoparticles. Okay, this is now, it is not done by any other field, right? That means that's why we are now focusing on the microencapsulation techniques, right? By doing the microencapsulation, we can enhance the bioavailability, right, without influencing or without changing the structural change. We will not do, right. Only by changing the means by encapsulation technique we can enhance the bioactivity. This is what I want to tell. Now here you can see here, this is you can see the Z isomer, right, carotenoid isomer. Okay, if we have Z isomer or cis isomer, okay, easily it is forming the nano suspension. But if it is a trans isomer, then it is very difficult to make it to nano suspension. That means it is a submicron, right? And this is the problem here. Now this is the area where we have to look. That means the challenge is that we should prepare the nano suspension and we should keep the molecule trans, right? Cis should not be there, right? And cis are actually converted into nano. There is no value because bioactivity is less here. Therefore, we have to make them, you have to keep the molecule trans, right? And we have to make the molecule into nano suspension. Okay, now this is one technique also we use. Okay, that means I will tell you in the astaxanthin case how we actually use, right, the nano emulsion techniques, right, or encapsulation techniques also we use, right. Particularly nowadays we use cyclodextrin, right, cyclodextrins, gamma cyclodextrins we use. Okay, particularly gamma cyclodextrins are used for making the molecules water soluble, and then after that increasing the bioavailability. Okay, and then also we can use PLA, PLGA, right, technique, okay, for the for enhancing the beta carotene bioavailability, right. But here is that the thing is that the cost effective, because if we are using a PLA, PLGA technique for preparing the formulation, then that may not be cost effective. It may be a research for research purpose, it may be good, but in case of commercial purpose, it is not actually viable. Therefore, most of the pharma industries, they switch to the cyclodextrin based carotenoids, right? And also they absorb the carotene into a porous starch molecules, and they make it water soluble. Now, this is the technique most commonly find in the laboratory, right, in the research lab, right, how we can prepare the emulsion of the carotenoids, right, lycopene. It may be a beta-carotene, it may be any other carotenoids, but lycopene is shown here, right. We can use T20, okay, and dichloromethane. This is important one, right. Dichloromethane we are using in the organic phase. But what happening is that the dichloromethane, when it come into with the because whenever this problem is that actually in the laboratory it is successful. It is, you can get the molecule, you can get the formulation easily in the laboratory. But if you want to evaporate the dichloromethane, right, it is in the last phase. You can see after evaporation of the organic solvent by rotary evaporator, you will get the product. But it is a difficult task in the industry. You cannot evaporate dichloromethane, right. This is the problem, because if we know that yes, this technique we can use for the preparing the formulation, but in the industry it is failed. Why? Because there is no evaporation technique for the dichloromethane, because dichloromethane you cannot evaporate by a spray dryer. And if we are using a rotary evaporator, dichloromethane residue will be there in the formulation, and it may not pass through the regulatory agency. Therefore, always the research, whatever we do in the laboratory, is not always applicable in the industry. That is the reason why. Therefore, if you have a new technique, that means without using dichloromethane, if you are able to use ethanol, right, as an organic phase, and you are able to make your preparation, that means if you are able to prepare the things, then it is good. Or if you are using a cyclodextrin, right, and you are able to prepare the water soluble carotenoid, then that is good, right. You cannot use this dichloromethane, and you cannot use this lycopene solution. For example, if you want to prepare lycopene eye drop, you cannot use this dichloromethane solvent or ethanol here, right. This is the problem here. Now we are doing research, we are developing new molecules, new dosage form, we are publishing the article, but there is no value at the industry level, because this is the problem we are facing here. Therefore, we have to think at the end. That means if it is not applicable, right, to the that means we are not able to transfer into the industry and to the market, then no value of doing this research. Only at the publication level we will publish, then we will forget it. No value at all. Therefore, we have to think a new way of doing the formulation, and still it is under research. I am not having this technique also, right. We are doing research, right, and still we are trying for a new method of microencapsulation, so that we can avoid the use of organic solvent while preparing the nano emulsion or nano suspension or any micro emulsion or anything. But CO2 technique is good, but the problem is that there is a formation of trans 2Cs, right. There is a problem there. Now, thing is that I want to tell here about the astaxanthin, right? But I have discussed a lot of things regarding other carotenoids, right? Now, this astaxanthin, already I told you, it is an oxygenated pigment, right? Having molecular weight of 596. If it is free astaxanthin, but if it is esterified, then the molecular weight will be always higher, right? You will get a different RT, you will get a different RF value, right, if it is a monoester form or diester form, right. And you can see the antioxidant capacity of this carotenoid is higher than the vitamin E, because all of you know antioxidant capacity of vitamin E is high, but you can see vitamin E having a lower antioxidant capacity than the astaxanthin, okay. And in compared to beta-carotene also, right, beta-carotene is having 100 times less antioxidant capacity than the astaxanthin. Right. Now you can see the value of this carotenoid. Why it is important to do more research on the carotenoid astaxanthin. Now it can cross blood-brain barrier, it can cross blood-eye barrier, and it can give prevention to neurological disorders like Alzheimer. We have done research on astaxanthin, right, and Alzheimer, and how it is actually preventing the Alzheimer disease. That also we published, right, in most of the journal, right. Almost all four publication we are having on the Alzheimer, right, and it is found that very good molecule. It is a very good molecule for preventing the Alzheimer disease, right, and also very good molecule for cardiovascular health. That means it is a good antioxidant. Because it is an antioxidant, it will always give good effect, right, and it can prevent the disease which are related to oxidative stress. Now the thing is that how we can make the astaxanthin natural way. That means you can see this is a algae, right, called Hematococcus pulvialis, right, and this algae actually gives best carotenoid, right, that is astaxanthin. You can see it having a two different phase, right. One is the green stage and another is the red stage. Okay, the red stage means in the green stage actually it is grown, right, under stress. Okay, when it is the cells are undergoing stress level, right, it converted into red color. That means the astaxanthin actually accumulated inside the cell. But the problem, but the issue is that why it produces carotenoid? Because what happens is that naturally when we give a stress to the cells, okay, it produces carotenoid. If you want to give stress like UV light, for example, if you give UV light, if you change the nutrient, if you change the pH of the solution, right, immediately we are giving the stress to the cells, and it will produce the carotenoid inside the cell in order to protect itself. In order to protect itself, it produces the carotenoid, that is astaxanthin, right. You can see how it is actually producing. Now you can see in the 0 day, that means fully grown under microscope, right, it will give a fully green colored. Then day 1, if you are giving stress, right, the carotenoid composition, means carotenoid accumulation starts, right, and then in the 4th day onward the carotenoids start accumulating, and at the 16th or 17th day you will find the carotenoid content is at peak level, right, and almost all 3% weight by weight of the carotenoids you will get in the algal cells, that is Hematococcus pulvialis. Right, now if you do the analysis of these carotenoids, right, you can see there are two different chromatograms, right, one is green and another is red. Okay, now you can see the green color carotenoids they are the carotenoids, but you will not find any astaxanthin here. Astaxanthin content will be very less. Okay, that means they are green, but once they are converted, that means astaxanthin is converted inside the cell, you will find the peak is changed. That means by observing the carotenoids peak, right, you can understand that yes, carotenoid is formed, that is astaxanthin is formed, and you can harvest the cells, right. And you can see here astaxanthin diester is there, beta carotene peak is there. Beta carotene peak, you can see there is a high beta carotene peak, right, but in the initial level there is a low beta carotene peaks. That means astaxanthin monoester is there, right, and then we have astaxanthin free form. Astaxanthin free form will be very less. Already I told you, if it is a natural astaxanthin free astaxanthin will be very less in the cell. All the carotenoids will be monoester form, right, or in the diester form. Therefore astaxanthin free form of the astaxanthin will be very less. Okay, and other carotenoid will be also there. Lutein will be there, zeaxanthin will be there, chlorophyll will be also there, little bit of chlorophyll, right, and astaxanthin monoester there. Okay, beta carotene is there, astaxanthin diester is there. Now this chromatogram will give you that how actually a natural carotenoid actually look like. But if it is mixed with a carotenoid, that means synthetic astaxanthin, immediately you will get a free astaxanthin peak here, large peak, right, sharp astaxanthin peak you will get here. That indicate that the sample is actually contaminated with the or adulterated with the synthetic astaxanthin. Okay, this is actually a new source that we actually worked in our laboratory in Jamia Hamdard, in School of Pharmaceutical Education and Research, in our laboratory, the microbial pharmaceutical biotechnology laboratory, and we have bypassed the algal source, because algal source gives astaxanthin, which are free form, carotenoid, that is a esterified form, but it taking too much time, right? And there is chance of contamination also, and the growth is also slow. Therefore, what we have done is that we used a yeast cell that is called the Papia rhodozyma. Okay, this yeast is actually able to produce carotenoid. You can see this is the yeast is grown in a YM agar media, that means yeast malt extract agar media. You can see the red colonies, right? There's a streak in the YM agar media, right? And then there are sec flasks, right, in a conical flask in presence of light source, right? And then after that, the sec flasks, our cells are harvested, right, by centrifugation process, and then we have extracted by using DMSO. Then you can see the red colored extract, right, which is actually actually a carotenoid having antioxidant, high antioxidant profile. Now similarly, we also grown the yeast inside a reactor in a 5-liter fermenter, right? And then we harvested the whole broth. Then after that, we centrifuge the whole mass, right? We get the wet cells. Then after that, we dry it by lyophilizer, right? Because you cannot go for tray drying, or you cannot go for any other spray drying here, because the carotenoids are highly sensitive molecules. They can degrade at high temperature. Therefore, we lyophilize it. And you can see, imagine here that when we lyophilize, the color is less dark. But the same cell, right, when we dry in a oven in presence of high temperature, right, it give a good red color. That means the color intensity is more. And if we dry by a freeze drying technique, the astaxanthin is there, but the color of the compound is light pink color. Why it is happening? This is the important area now. Now what happens here is that I can tell you the reason why it is happening. Now whenever we heat a carotenoid, right, it gives a bright red color, right? But if we don't heat, okay, it gives always a dull pink color, okay, or it gives a less red color, right, less dark color. Now you can do it in the in home also, a simple experiment. Now if you have a prawn, right, just you can heat it. Now if you heat a prawn, right, by if you boil, or if you just you give some heat, right, to the prawn, what happens here? That the color of the prawn actually changes to red. Okay, initially the prawn color is white. Why? Because the carotenoid is always in association with the proteins, right? Now once they are associated with the protein, they are always dull color, right? The color of the carotenoid is always whitish color. Okay, the pink color will be there, or whitish color will be there. Once you heat, the protein denatured and it free from the astaxanthin or any carotenoid, right? And then the color became dark, right? Therefore, always when you give heat, okay, always there is a increased color intensity of the carotenoid. This is also one test whether the product is containing carotenoid or not, right? For example, if we give some heat to the carotenoid containing material, intensity of the color will always enhance. Okay, this is the reason why the color is dark, because same product we have prepared in a hot air oven, right? And same product we prepared in a freeze dry. Now we can, in the freeze dry there is no heating, therefore the color is dull, right? But if we are doing heating, right, and we are heating at a high temperature, then immediately the color will increase and the intensity of the color will enhance. This is the flow of astaxanthin production, right, by carotenoid, right? This is the seed culture of the microbe, right? We do fermentation, cell harvesting, then we do cell cracking by physical method, ultrasonication, right? Or we can use DMSO also. DMSO, preheated DMSO we can use to rupture the cells, right? And then after that we dry. But here the cell harvesting we can dry also. Same thing we can do. We can extract by a chemical method like DMSO, all the things, right? And then we have to stabilize it by using vitamin E. We can use BHA, butylated toluene, right? BHT, BHA. Okay, any antioxidant you can use. Vitamin E also you can use. Vitamin C also you can use. But vitamin C is not prepared here because the oil soluble. These carotenoids are oil soluble. Therefore, vitamin E is most commonly used stabilizer. Okay, antioxidant. Okay, for stabilizing the carotenoids like astaxanthin. But if we made into a water soluble compound, right, then vitamin C we use, right, as a stabilizer. But if you use a vitamin E in the water soluble carotenoids, right, no value at all. Therefore we have to think whether the product, whatever the end product you are getting, whether they are oil soluble or whether the water soluble. If it is a water soluble, then use vitamin C as an antioxidant. If they are oil soluble, use vitamin E. Tocopherol you can use as a solubilizing agent. Then you have to store it at low temperature. Okay, apart from, means away from light, right, and temperature below room temperature. That means around at 20 degrees centigrade or 4 degrees centigrade or minus 20, depending upon. If you are storing at minus 20, then you can store up to 2 years. This is the spectral data, right, of astaxanthin. You can see a trimexima here. And these are the mass spectra of carotenoid, that is astaxanthin. We have to do LCMS, otherwise no value, right? Therefore, at the end we have to do mass, because you can see this is my student name, Muzir. Okay, actually he has done research under me, right, PhD under me, and Muzir Hassan, his name, right? That's why the name come here, Muzir, right? And this technique actually we transfer to the Chinese industries. Now they are producing the molecule. And this my student Muzir Hassan, actually he visited China. He stayed there, right? And we demoed the process in their laboratory, in their reactor. Then after that they agreed to take our technology. Right now, these are the mass spectra of the carotenoid, because once you have mass spectra data, then only you can have a clear indication that it is producing the carotenoid. Now you can see 597, right? You can see 812, right? You can see 1057. These are the actually molecules. They are monoester form. They are the diester forms, right? And this is the ester form. And some of the compounds you may get which may be degraded, okay, during the mass spectroscopy. Okay, now thing is that we have also done some of the things like related to formulation, right, on astaxanthin, right? And we prepared astaxanthin. And then after that we prepared the astaxanthin formulation. Then we tested in the animal models. Then we have done the gamma scintigraphy imaging, how the biodistribution is carried out, right? And then also we tested the in vitro study by using the PC12 cell line, right? And how actually it is preventing the neurological disorders, right? And that we have actually carried out in our laboratory. And then this is the things. Actually I'm not discussing these things, right, because these things you can read the article I publish also. I can show you at the end, okay, the publication, so that you can go through the publication, or else you can ask me, right, how actually we do the astaxanthin solid lipid nanoparticles actually for enhancing the bioactivity of the astaxanthin, right? These are the things actually we do. You also do, because therefore I'm not going in detail about these things, right? How we can prepare the solid lipid nanoparticles. Very simply we do it by using the polaxamer, because I already told you nano formulations of the carotenoid is very important. because you need control release stability, okay, and lipophilic astaxanthins are there. Therefore, how we can improve the delivery of the compounds, right? Then we have how we can plan the molecules. That means we can use the surfactants like polaxamer 188 and lecithin. Also we have used solvent, we have used dichloromethane. Again, same problem. I already told you dichloromethane is a problem. Still we have used dichloromethane, but we don't know till now. We are having a problem that how we can overcome this solvent. That means without using dichloromethane, how we can prepare the nanoparticles, right? These are some of the things already we are doing. Therefore, I'm not explaining it in detail, right? Because optimization, all of you know the response surface methodology, and actually we started response surface methodology work in 2005. At that time, these are the very new techniques at that time, and then we propagated the technique. I used to tell other also in Jamia Hamdard. If you see, most of the publications on response surface methodology appear in the journals around in the year 2006, 5 and 7. Okay, up to 2012 it was a peak, right? Then after that everybody used these techniques, and now it is a common technique for the of using this box Venken design or central composite design, right? But still we have newer techniques, right? That is need of our need at the hour, right? That means genetic logarithm or physiologic or neural network. If we are using these newer techniques, then you will able to publish a new articles, new things, right? But response surface methodology is now old, and now the journals are not preferring RSM techniques, right? And these are the techniques already you know. Therefore, I'm not going in detail, because optimization of the process and the formulations we have to do by RSM. Then after that we have to validate it. Okay, means the formulation validation we have to do. We have validation can be carried out in terms of particle size, PDI, okay, and then the release also very important. Okay, in vitro release of the astaxanthin solid lipid nanoparticles. Then we have to have the size and the microphotographs, SAM and TAM. Everything is very important. See, this work we have done in 2012. That means you can see at that time it was a new research, but not now, right? It is at that time in 2012. These are new techniques, but now if we do this work, this is a old, outdated work. Okay, therefore we have to do newer work, right? How we can prepare this carotenoid into eye drop. Right, this is the things we have to do. We have to think. Then we have to do the biodistribution study, because once you have given to the animal, it is not that it is bioavailability study. You have to do. You have to study the biodistribution study. Then for that you have to use 99 TC99M. Okay, this work we have done in DRDO, right, in Delhi. And then because we don't have the lab or facility to handle the radio level substances, okay, therefore we have done the experiment there in their laboratory in DRDO, okay, in mass, right? And then we find out how the bio distribution of the nano formulations or solid lipid nanoparticle of the astaxanthin is happening inside the rat. Okay, that means we can we have given intravenous route, right, intranasal route, right, and then we have find out how actually it is accumulating at a different organ. Right, these are some of the data where it is actually accumulated, right. Now we can see intranasal route, right, or by injection, how we can change the change the carotenoid distribution in the organ. Right, this is the intravenous route. You can see the change here, right? Intranasal route, okay, and intravenous route. Now, intranasal route always better. Therefore, we publish the article how we can prepare the astaxanthin directly. We can deliver into the brain through nasal route. I can show you, right? See, gamma centrography image of the animal, right? And there is a good distribution of the astaxanthin, right, in the brain, because we are targeting there at that time how it can protect the neuron, right? And therefore we have always seen how actually we can formulate the product so that it can transfer into the brain, right, through intranasal route. Now, then after that, we have done some of the activity on PC12 cell line, right, through the hydrogen peroxide induced oxidative stress, and how actually because we have to induce the oxygen stress, okay, oxygen stress or oxidative stress we have to induce by hydrogen peroxide, right? This will cause the damage in the neuron, right? And then how the astaxanthin actually protecting the neuron from oxidative damage, right? Then we have to see the glutathione level, right? Now you can see how actually happening, right? This astaxanthin, okay, how actually it is preventing that means oxidative stress by observing the glutathione level, right? The glutathione level is always decreased in case of oxidative stress, but if we give the astaxanthin, actually it increase the glutathione level and it prevent the oxidative stress. Lipid peroxidation, right? You can see lipid peroxidation level is increased, right, in presence of hydrogen peroxide, right? But if we treat, then lipid peroxidation is decreased, right? These are the some of the experiment actually we do normally in the laboratory. You also do, right? This is not new things, right? Whenever you have a formulation, we have to prove that how it is actually efficacy is there, okay, under in vitro or under in vivo condition, right? This is somewhat conclusion of the PC12 cell line we have used. Now this study we have extended now in the real Alzheimer disease, right? And now we have published article in Alzheimer research, right? And how actually the astaxanthin is actually preventing Alzheimer disease, and how actually it is preventing the this amyloid beta plaque, right, in Alzheimer models, okay? But that work I am not showing because it's already time is almost all much time I have taken, right? But if you want, then I can share my articles to you so that you can read and you can understand that how we have done the research, right? And this is the technology we actually transfer the astaxanthin fermentation, astaxanthin process to a Chinese industry, Global Biological Corporation. And this is the he's the CEO of Fujiwang, right? He's the CEO of this company, and he is our ex vice chancellor. But now he's there in the IIT Delhi as a professor emeritus, but he was our vice chancellor at the time. And this is the what we achieved in our laboratory. And so these are some of the old publications on astaxanthin. Okay, we published in 2012, and Prakash Bhatt, okay, he actually carried out some of the fermentation process on astaxanthin. We published in Biocatalysis Agricultural Biotechnology, right? And then we have Mujir Hassan, right? He worked on astaxanthin extraction. I already told you this is also one area how we can use different technique like ultrasonication, supercritical fluid extraction technique, high pressure homogenization technique, how we can use these techniques for extracting the carotenoid from the cells. Mujir Hassan and other fellow also, they worked and they published it in Preparative Biochemistry and Biotechnology. Right, and then the work I have shown here, that is the REC Advanced, okay, how we prepared the astaxanthin formulation and how nano formulation, how we prepared, and then it is actually targeted to the brain, right? That part we have published along with the student Prakash Chandra Bhatt, right? And this is my laboratory, okay, in Jamia Hamdard, New Delhi, where we do a lot of research on nutraceuticals and plant metabolites, phytomolecules, phytochemistry, right, purification, fermentation, bioprocess, biotransformations, right, formulations of new molecules. Now we got a project from Canada based on carotenoids, how we can develop the carotenoid into eye drop that I was telling to you, that of value of around 75 lakhs Canadian dollar, around it is 1 lakh around Canadian dollar, that we got project, and here the task is that how we can prepare the carotenoids, formulate the carotenoids into a suitable eye preparation so that it can prevent the age-related AMD, means age-related macular degeneration, right? That project I got recently, right? And I'm working on these, how we can develop the carotenoid into next level. And further also we are working on lutein extraction and zeaxanthin extraction from the algae, and hematococcus. We are also working on hematococcus. Okay, a lot of carotenoids we do, right? And also there is a scope also in Orissa or in your part, right? We have orange flash sweet potato, but we are underutilized. That crop is underutilized. That means we are unable to utilize that for beta carotene extraction, right? But there is a scope here that we can use those tubers for extracting the beta carotene, okay, and you can use it for the nutraceutical purpose. And this is the group, right? And this is the old group actually, and this is myself, right? And he is Mujir Hassan, okay, actually worked on the astaxanthin, right? Azhar, okay, and he is Prakash Bhatt. Okay, now he opened his own company in IMT Manesar. He's doing good business in nutraceutical industries, right? He is placed in SSP Faridabad. He's in R&D as a R&D manager. Azhar is placed in Cincinnati University, USA. He's doing research on anti-cancer molecules, right? And Pooja, he's now in the UK, right? And most of them are there placed in the outside India and doing good work. Right, thank you all of you, right, for your patience and listening to my lecture on carotenoids and particularly astaxanthin, and thank you very much for your kind attention.
Thank you, sir. Nice presentation. So you have enlightened so many things related to your herbal drug formulations. So I think it will be very useful for the faculty members and also the research scholars who are involved in this area. So we have received some questions on chat box, sir. Please, we can check it.
Can I check your comments, right? Yeah. Yes. Yeah, one question I'm getting, that is it possible for prepare a metal complex with natural drugs for anti-cancer activity like curcumin? right? Yes, it is possible, but we have to think a reactive. But it is possible in case of nicotine. I think you understand that nicotine, we can prepare the zinc compounds. We can prepare particularly zinc compounds, right? And also we can prepare because zinc is very important molecules if we consider the curcumin, right? Because curcumin having antioxidant activity, anti-diabetic also, and if we are able to make a complex with a zinc, right, then it will give you more effectiveness. But still the chemistry part is required to do lot of research on preparing this, and also we have senoside. Okay, we have senoside with combined with the metal ions, right? And similarly we can try, but still we need to do some research on this area, how to prepare the this metal based phytomolecules. May create toxicity, that has to be evaluated, right? It may create toxicity also. That means if we prepare the metal complex of any of the natural molecules, right? But if we are using a metal like zinc or similar compounds, then it may not cause any selenium, then it may not cause any toxicity. Yeah, that we can use in the ocular formulations, but still we have to prevent this the use of solvents, right? Solvent, but now we don't have that method to prepare the ocular formulation for the astaxanthin or any of the carotenoids, because the bioavailability issue is there. We can use penetration enhancer, right? And we can use some of the surfactants here, right? But still there is a issue of presence of organic solvents, right? And there is an issue here, right? How we can prepare the ocular formulations of the astaxanthin and other carotenoids. Yeah, any questions directly that you can ask me also? Yeah, flavonoid content, we can we confirm flavonoid content of extract by using UV spectroscopy? Yes, we can measure flavonoid content, but it will give you total flavonoid, right? We cannot measure the flavonoid chromatogram. That means what are the flavonoid is present. Okay, for that we have to do this LC, SPLC. Green method, we can use solvents like I can tell you that green solvents, actually they are I've forgotten the name, right? Actually we have green solvents, but actually I'm forgetting the name of the solvent here. We have some of the green solvents actually use, but that is under research only, right? Like limonene like molecules actually use citral, limonene, and also solvents like I'm forgetting the name. I can send you the mail, right? I'm not having the microwave extraction also suitable, but it is only on this, right? We can go for the large scale production, but ultrasonication is nowadays picking up, right? For extracting the molecules by the extracting the molecule from the plants, ultrasonication techniques. Chitosan based nanoparticles actually used, but actually the stability issue is there. Therefore, whenever we are using chitosan, it has to be cross-linked with the TPP tripolyphosphate. Then only you have a stable chitosan nanoparticles, right? And we can use, but the acid degradability or acid degradability is still there with the chitosan nanoparticles. Therefore, if we have a chitosan nanoparticles, we have to have a strong cross-linker like tripolyphosphates.
Thank
Yeah,
you, sir.
flavonoid. Yeah. Now there is a question: what is the effect of temperature on flavonoids? Right.
Yes, yes.
Flavonoids are highly stable molecules, and they are not degraded by temperature if you are going beyond 70 degrees centigrade. Right. Therefore, spray drying techniques are mostly used, right, for flavonoid drying. Right. Mostly the spray dryer of temperature around 60 degree. Inlet temperature is 120, right, and outlet temperature is 60 degree. Right. That is most preferable temperature for drying the flavonoids. And maximum we can go up to the 70 degree centigrade.
Thank you so much, Dr. Bibu Prasad Panda, sir, for sharing your vast research experience and importance of various natural resources in the benefit of our healthcare. We remain most grateful to you, sir, for the rapt attention with which you have delivered this talk. May I now request our principal, Professor B.V. Ravi Kumar, sir, coordinator of this FDP, to present an e-certificate of appreciation to Dr. Bibu Prasad Panda, sir, as a token of our respect and affection from entire RIS fraternity.
First of all, I would like to thank Dr. Bibu Prasad Panda, sir, for accepting our invitation and coming forward to share his knowledge. And I would like to congratulate him for doing good research. Please continue the good work you are doing, and it will be definitely helpful for the society. And you have delivered a good talk. I hope all the participants will be benefited by your lecture that you gave today. Thank you, sir. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Kindly accept it, sir. Thank you, sir.
Thank you.
Thank you very much, sir. Dear participants, with this, the session 1 of Day 3 FDP and FDP comes to an end. We'll be resuming today's second scientific session at 2 p.m. exactly on the same platform. Till then, please stay safe and stay connected with us.